And it's my pleasure to welcome Dr. Aaron Smith back to Madison today. As some of you may know, Aaron obtained his PhD in chemistry here at UW-Madison in 2012. Seems just like yesterday. His groundbreaking research in Judith Burston's lab, who's also here today, focused on the chemistry of heme ligands as regulators of protein activity. And I still think it's some of the most important work in this area. From there, he went to Amy Rosenzweig's lab to study the structural biology of a cadmium, cobalt, and zinc transporter. In 2016, he moved to University of Maryland to set up his own independent research lab. But there he saw the light to return to studying iron, my favorite element. And now he's combining biochemistry, structural biology, and inorganic chemistry to study several iron-dependent mechanisms in transcriptional regulation, protein modification, and transport. And today, I believe he'll tell us about some of that work on ferrous iron transporter from bacteria. That's really quite fascinating, I think, and is broadly conserved across the bacterial world, as well as this new interesting iron sensing two-component transcription system. He's been extremely successful at Maryland. He holds an NSF Career Award as well as other awards. He's a passionate mentor, and he's now program director of Chemical Biology Interface Training Grant. It's quite a lot. And his research program has been well funded. So I want to welcome Aaron back to Madison.
All right, well, thank you all very much for being here. It's really a pleasure to be back in Madison, and it gives me an opportunity to use what I'm sure is a completely verboten use of Bucky's image here, but was very common when I was a graduate student. I'm happy to be here today to tell you a little bit about some of the work that's been happening in my lab at University of Maryland, Baltimore County, or UMBC. I just wanted to first start off by telling you a little bit about UMBC because maybe you don't all necessarily know about us. And if you do, it's probably because of this basketball game that happened about 7 years ago where UMBC defeated #1 Virginia in the NCAA tournament. But we are more than just a university that occasionally has a good basketball team. We are a medium-sized STEM-focused R1. We've got about 15,000 students in a suburban setting. We're outside of the city of Baltimore, and if you squint really hard, you can hopefully see the skyline of Baltimore right there. This is part of our main campus. And at UMBC, we really have a major emphasis on inclusive excellence, which is this idea that diversity is absolutely critical to our success as a university, and that we aim to serve a diverse population, which is the population of the state of Maryland. This is really exemplified by a number of programs at UMBC, including the Meyerhoff Scholars Program, our GNRU Rise programs, as well as a variety of different postdoc fellowships. Proudly, I think these have been quite successful, allowing us to be the #1 producer of STEM undergraduates who are African-American who go on to get MD PhDs in the United States. And we've also been able to close GPA and graduation gaps, not only for underrepresented individuals in STEM disciplines, but also for non-STEM disciplines. So my lab is located at UMBC at this campus, and it focuses on iron because it's a bioinorganic lab. We are interested in how inorganic elements function in biological systems. And I would argue that there is no more important inorganic element than that of iron. And even if you disagree, certainly nature knows that iron is special, having adopted iron to serve in indispensable biological processes from DNA biosynthesis and aerobic cellular respiration to a suite of metabolic processes that are responsible for keeping everyone here in this seminar alive. So my lab is really structure function focused, trying to understand the structure of enzymes that utilize iron. We have one major project in the lab that I won't talk with you about today, which is trying to understand from a eukaryotic standpoint how a post-translational modification known as arginylation, which is a really critical post-translational modification that regulates how long or how short-lived proteins can be within the cell, how that is ultimately regulated by the presence of iron-sulfur clusters, as well as the presence of oxygen. I won't have a chance to talk with you about that today, but if you're interested in that, I'm more than happy to talk with you after this seminar. Instead, today what I'm going to focus on is a prokaryotic system. And in particular, the motivation for the study of this has to do with antibiotic resistance. Now, this is a problem that we probably don't think about as much in the United States as we should. And this is probably motivated by the fact that highest rates of antibiotic resistance, these are oftentimes observed in Southeast Asia and other developing nations. But this is not a problem that we are immune to. About 2 million individuals in the United States are infected by pathogens that are antibiotic resistant per year in the US, translating to about 40,000 deaths per year. But hopefully, as you can appreciate with these graphs here on the right-hand side, we're seeing major increases in antibiotic resistance, meaning that those number of deaths are going to increase. Now, here in the US, the two types of bacteria we have to most commonly worry about are antibiotic-resistant C. diff, as well as carbapenem-resistant Enterobacter. But that is just for now, because we know our world is global and interconnected. And just as we can get on airplanes, bacteria that are antibiotic resistant can also travel on airplanes. Furthermore, in the United States, we shoot ourselves in the foot in a variety of different ways, but that also includes overuse of antibiotics in humans when they're completely unnecessary, as well as overuse in commercial agriculture. And I think this point here, this last point is maybe the scariest, which is that most of the most serious antibiotic-resistant infections in the US are nosocomial in nature, which is to say that you might be sick or injured, you go to a doctor, you go to a hospital, and you may come back from that location with an infection worse than the injury that you had in the first place. So that's a problem. Now, where does iron come into this? Well, most bacteria require iron in order to establish and sustain infection within a human host, and they have to get iron from us. This has led multiple labs to suggest that perhaps targeting iron acquisition pathways might be a way to combat some of these virulent strains of bacteria. Now, if you put yourself in the shoes of a bacterium, you've got a couple of different oxidation states of iron that you ultimately have to acquire. The first is what's known as the ferric form of iron, or iron in the +3 oxidation state, but it really represents some problems for bacteria. It is generally both chemically as well as kinetically inert, chiefly because it's highly insoluble under physiological conditions. It spontaneously forms iron oxides and iron hydroxides. Now, outside of a human host, that's no problem. It's highly environmentally prevalent, and you go outside, you'll dig up plenty of iron oxides and hydroxides, but it's a problem within the human host because it's hard to get. Moreover, it's a hard Lewis acid, and what this means is it's not very versatile. It preferentially binds to nitrogen and oxygen-containing biomolecules. In contrast, the ferrous form of iron, or iron in the +2 oxidation state, is much more attractive to bacteria. Certainly, it's more chemically reactive, oftentimes doing one-electron chemistry in the presence of oxygen. That certainly has to be controlled. But it's also more kinetically labile, which means it can be transported more readily within the bacterial cytosol. It's also highly soluble under physiological conditions, but it is less prevalent in the human body. However, I'll come back to that point and talk about some niches where it's highly dominant. And finally, because it's a borderline Lewis acid, it actually binds to nitrogen, oxygen, sulfur, and phosphorus-containing biomolecules, which makes it more versatile. So if you're a bacterium, you'd really rather acquire and transport this form of iron. But historically, we've studied a lot more about how bacteria acquire this form of iron. In fact, we know enough about these processes, although I would argue that we need to know more. But we know enough that we can draw these cartoons here on the left-hand side, but those represent atomic level structures that we have, as well as mechanistic information that we have about how iron is trafficked, for example, in iron protoporphyrin 9 or heme, as well as how it's solubilized and trafficked in ferric siderophores. By contrast, however, we know a lot less about iron 2 homeostasis. And this is rather unfortunate because there are a number of niches within the body, either perfectly healthy human or a diseased human, in which iron 2 might be prevalent. This includes under conditions that might be highly acidified, like parts of the stomach and even parts of the intestines. locations where there might be low oxygen tension, like parts of the intestines, as well as anywhere where a bacterium can form a biofilm. This can be skin of burn victims, lungs of cystic fibrosis patients, as well as if we forget to brush our teeth at the interface of our teeth and our gum lines. Now, we're fortunate that there are a number of model studies that have shown that iron 2 actually contributes really strongly to the virulence of a number of bad pathogens, from those that cause nosocomial infections to causative agents of Legionnaire's disease, cholera, and even the most common infection worldwide, which is gingivitis. So this suggests if we had a better understanding about how this is trafficked within the bacterium, we might be able to target this pathway. So what do we know about this pathway? Well, the predominant prokaryotic iron 2 transport system is what's known as the FeO system. This was first discovered in E. coli, and this is an exaggerated illustration of what that operon looks like in E. coli. Unsurprisingly, downstream of binding sites for 2 iron-responsive transcriptional regulators are 3 open reading frames for 3 proteins, FeOA, FeOB, and FeOC. FeOA and FeOC, these are small, soluble cytosolic accessory proteins. And FeOB, this is really the major protein here. It's a wholly conserved integral membrane protein that's responsible for the translocation of the cation in the first place. So there's a lot that we didn't know after this system was discovered, but it was imagined that it occurred something like this. We're under conditions in which iron 2 is stabilized in the environment, or even in which it's not, because we know bacteria can secrete a wide array of different small molecules or redox active. They can affect the redox balance outside of the bacterium. That more labile iron 2 in a gram-negative bacterium is illustrated here, can diffuse across an outer membrane porin, and then in a manner that we totally don't understand, traffic to FeOB. FeOB gets iron 2 across the lipid bilayer. This is responsive to the presence of nucleotides in the cytosol, but whether this is an active transport process or facilitated transport process is unclear. And then what these little proteins are doing here in the cytosol was also unclear. So over the last several years, my lab has been trying to piece together the different bits of this system to understand in better detail about how it functions. Now, the major mystery here is the FeOB protein. And unfortunately, we still do not have an atomic level structure of this protein. But the FeOB protein is composed of a few different domains. It's a polytopic transmembrane protein with multiple transmembrane alpha helices. It has a small periplasmic domain that's rich in carboxylate residues that's sticking extracytoplasmically. And then it has an additional N-terminal domain that's really been the study of this system ever since it was discovered. This is referred to as NFeOB, and this has been chiefly studied because you can lop off this domain, you can express and purify it, and you can characterize it. So my lab, as well as others, we've structurally characterized this protein, not only in its isolated form, but also throughout its entire catalytic cycle. In fact, this whole catalytic cycle was actually derived by a former postbac in my lab, Kate Magante. And what's interesting about this protein is that it has structural homology to eukaryotic G proteins. And in fact, when you look at its nucleotide binding and hydrolysis capabilities, it most commonly will bind and hydrolyze GTP, but it also does ATP in addition. But there's a problem with this, which is if you look at how quickly this domain hydrolyzes GTP, it does it incredibly slowly. About 1 GTP molecule hydrolyzed every 10 to 15 minutes for this domain in isolation. So if this is active transport, this rate of GTP hydrolysis is completely incompatible with active transport. So we started thinking, okay, we need to think about the intact protein and ask the question whether the intact protein functions in the same way. And when we got started working on the intact protein, there were very little information out there in the literature about how this might function. In fact, all there really was a homology model here that was generated based on a crystal structure of just the NFeOB domain that was suggestive that a pore could be formed at an obligate trimer lined with cysteines, and this was the location for iron 2 to get into the cytosol. But there was no biophysical evidence for this. So we wondered, could we characterize that and get a sense of whether or not this model is accurate. So this was something that was worked on by a former graduate student in my lab, now Dr. Alex Sestock, where she really took a tried and true structural biology approach. She cloned, expressed, and purified a variety of different intact FeOBs from a variety of different organisms, some more compact than others, some with extra domains, some with fewer domains, ultimately with the goal to structurally characterize this protein. But I think as any of you who work on membrane proteins can appreciate, you can't just get expression and then you're off to the races. There's a lot more that you have to do after that. So for example, if you need to membrane isolate, you have to figure out how to do that and keep the protein stable. If you decide that you're going to take the protein out of the membrane, like for example, using a non-denaturing detergent, you have to figure out how that will work and be compatible with your protein, as well as trying different lipid combinations and different solubilization conditions. So Alex slogged through this, and ultimately she could show nicely that we could get good yields of the intact protein now to allow us to start to ask biophysical questions. So one of the first questions that she asked is simply, what do we see in solution for this protein? Do we see any evidence for stable trimerization? First, looking in the detergent solubilized protein, we never saw any evidence for stabilization of a trimer. In fact, using analytical gel filtration as well as blue native page, all we ever saw was either monomeric protein or completely aggregated protein. Now, the aggregate is not uncommon for membrane proteins. You pull them out of a lipid bilayer. If they're incompatible with this detergent, things can get messy really quickly. What she showed for multiple different FeOBs intact in multiple different detergents was chiefly monomeric. Now, there's a problem here though, because we've got a detergent. That detergent is surrounding that transmembrane domain. That could very easily disrupt oligomerization. So then she asked the question, well, what happens if we put the protein back in a lipid bilayer? So she used a nanodisk approach for this, orthogonally expressing and purifying a membrane scaffold protein, then taking the membrane protein, the detergent, removing the detergent, and you get spontaneous intercalation then of the membrane protein back into the lipid bilayer. Again, using analytical gel filtration, separating out those empty nanodiscs, she again showed that this is chiefly compatible with only monomeric FeOB being present. But both of these have drawbacks because they require the use of the detergent in the first place. So another former graduate student in my lab, now Dr. Mark Lee, he asked the question, what happens if the protein never actually sees a detergent in the first place? Instead, what he used were these recently developed styrene maleic acid copolymers that can function almost like cookie cutters and can break membrane proteins directly out of native lipid bilayers. And then if your membrane protein is tagged, you can then purify your protein, and it's never seen the detergent in the first place. He then used mass photometry now instead of analytical gel filtration, and again was able to show using mass photometry that it appears to be chiefly monomeric in solution. Now, we tried to crystallize this protein. We were only partially successful, so right now we're trying cryo-EM with our nanodisk embedded as well as our small embedded protein. So we don't have a structure of this protein yet, although we're working on it. But these approaches still suggest that the protein appears to behave as a monomer, which has led us to consider, is there a different model then that might be existing for this protein? So we are fortunate to at least live in an age where we can ask AlphaFold, what does AlphaFold think that the protein might look like? And there are multiple different AlphaFold models of FeOBs that now exist. Here's just one example. This is the E. coli FeOB AlphaFold model here on the left-hand side. Here are those transmembrane alpha helices in the lipid bilayer. That the periplasmic loop that's up here appears to be actually bent over, at least in this model, on top of the transmembrane domain. And then here's that NFeOB domain for which we have a number of different crystal structures. When we look a little bit closer at that model, we see a couple of things that are interesting that suggest how the protein could potentially function monomerically. First, that periplasmic loop, highly electronegative, appears to funnel into that transmembrane region. Then, when we look deeper into the transmembrane region, we see a slew of cysteine as well as methionine residues that are present. Now, most of those methionine residues are probably holding together that alpha helical bundle, but the cysteine residues, 3 of them, are highly conserved, and appear to be at kinks in these alpha helices, very similar to P-type ATPases that transport copper and zinc as well as manganese. So right now we're thinking that a sulfur-rich pathway may be involved in the translocation of the iron 2 cation for FeOB to function as a monomer. So we believe for this to function as a monomer, those residues are going to be responsible for coordinating and translocating the iron in the +2 oxidation state. So this is something a current graduate student in my lab, Jeannie No, is working on using both the wild type form of the protein as well as selenomet substituted form of the protein. Also teaming up with Professor Kelly Chacon at Reed College using X-ray absorption spectroscopy to characterize what that iron 2 binding site looks like in that transmembrane domain. So that gives us a little bit of an idea for how this might be operative, and I'm going to return to that in just a few minutes. But it doesn't answer this question that was posed earlier with the N-terminal domain that's incredibly slow in its hydrolysis. So we then returned to the intact protein, asking questions about how does the intact protein bind and hydrolyze nucleotides? And Alex noted that in detergent, she saw a significant increase in the rate of nucleotide hydrolysis for the intact protein compared to that N-terminal domain. Now, this is still a fairly slow rate, about 0.1 per second, but virtually all of the FeOBs that we examined when they were intact had a much faster rate of nucleotide hydrolysis compared to that isolated domain. However, she did note that there were differences depending on the different types of detergents you use, like for example, whether you use a multicide or an ether-based detergent. This does affect the rate of GTP hydrolysis, suggesting that there are some important protein-protein interactions that are responsible for helping with this process. Now, does detergent negatively affect the rate of nucleotide hydrolysis? Well, Mark then asked the question for his SMALP encapsulated FeOBs. He looked at GTP hydrolysis, he looked at ATP hydrolysis, and he got similar KCAT values. So it doesn't appear that the presence of the detergent for the intact protein has a dramatic effect on the order of magnitude of that rate, which is much faster than just the N-terminal domain, but it's still slow for active transport. So we started wondering then, is it possible there might be some additional protein that might be present that can regulate this process? Maybe increase the rate of nucleotide hydrolysis under certain conditions or decrease it under other conditions. In eukaryotes, such as ourselves, we have this same problem. We've got plenty of G proteins. Some of them don't bind GTP very well. Some of them hydrolyze GTP really slowly. And so we use accessory proteins like guanine nucleotide exchange factors or GEFs, as well as G-activating proteins or GAPs to alter these rates and to increase the ability for these proteins to function. So naturally that begs the question, going back to the operon, is what are the roles of these other proteins like FeOA and FeOC in the operon? And could they function as GEFs or GAPs to help regulate this process? I'm going to tell you a little bit about what we think about how these proteins function. And I'm going to start actually with the FeOC protein because, spoiler alert, it doesn't appear to function as either a GEF or a GAP. In fact, it looks like a transcriptional regulator. It has a winged helix motif, as shown here on the right-hand side, with a helix-turn helix coupled to an unstructured wing region. And within that region are 4 cysteine residues that are highly conserved that we have shown can bind iron-sulfur clusters. And they don't just bind iron-sulfur clusters, but those iron-sulfur clusters are highly oxygen sensitive. So we've characterized this using a variety of different spectroscopies, EPR, X-ray absorption, electronic absorption as well. And when we do controlled reaction of this protein in the presence of oxygen, we can see rapid conversion of the 4-iron-4 to a 2-iron-2 sulfur species. Now, this is highly reminiscent of another transcriptional regulator known as FNR that binds DNA and also responds in a very similar manner. But unlike FNR, FeOC does not bind DNA as far as we can tell and as far as we have tested, and it does not dimerize, unlike FNR. And this is irrespective of whether it has or it does not have its iron-sulfur cluster. It appears to remain monomeric in solution. In fact, instead, FeOC appears to interact with the N-terminal domain of FeOB. There's a low-resolution crystal structure that suggests FeOC interacts in between these two key switch regions, switch 1 and switch 2, responsible for helping facilitate nucleotide hydrolysis, as well as respond to the presence of nucleotides. And we've used nuclear magnetic resonance to show indeed intact FeOB will bind FeOC, and these appear to be very similar binding locations as suggested in the low-resolution structure. However, if you take NFeOB, you add FeOC, you see no difference in terms of nucleotide hydrolysis within statistical significance. Moreover, FeOC is just simply not highly conserved. While this was the first kind of operon that was discovered in E. coli, only about 20% of bacteria have FeOC in the first place. Most bacteria have operons that look like this, in which FeOC is gone. But that doesn't mean it's fully dispensable. So if you delete it out, this is nice work done by Shelley Payne's lab at UT Austin, you still see problems with the ability of bacteria to grow and to acquire iron. So it's not fully conserved, it's not fully dispensable. What's it do? I'm gonna come back to that as well in just a moment, but at the time, we had no idea what it was doing. Instead, we thought that FeOA might be one of these essential partner proteins, this GAP or this GEF, based on a variety of biophysical evidence that we were ultimately able to garner. First, it is highly conserved across FeO operons. In fact, if you delete FeOA, you see a phenotype of as negative as if you delete FeOB itself. Moreover, insofar as structure and function are linked to one another, its structure suggests it's involved in protein-protein interactions. So we, as well as other labs, we structurally characterize FeOA. It has this small SH3-like domain, which is a small beta barrel, very commonly utilized in eukaryotes to facilitate protein-protein interactions. That is supported by some in vivo results, but then if we try to recapitulate this in vitro and pull down FeOA with FeOB, it looks like the interaction is highly weak. But these are under conditions in which nucleotide is not present. So we started modeling this interaction, and we noted that the model suggested that the interaction between FeOA and NFeOB occurred in the vicinity of the nucleotide binding pocket. So we started to wonder, is it possible that this interaction is actually mediated by the presence of nucleotide? So to answer that question, Alex, as well as a former undergrad in the lab, Sean O'Sullivan, who's now a neuroscience PhD student at UMBC, what they tried to do is leverage these naturally occurring fusions that are very common in the Firmicutes and the Bacteroidetes, where FeOA and FeOB are genetically encoded all together with a polypeptide linker in between. And what they did is they clone, express, and purify the intact FeOAB fusion as well as the N-terminal FeOAB fusion. They could get gobs and gobs of that N-terminal FeOAB fusion. It was screaming to be crystallized. We tried to do that throughout the entire catalytic cycle. We always got beautiful crystals. They always diffracted beautifully, and they always contained just FeOA within them, which is unfortunate. That flexible linker region is highly prone to proteolysis. So we got the structure of FeOA, but that didn't tell us really much of anything that we didn't already know. So we had to go a different route to characterize these fusions. First, by modeling the nucleotide-free form of that fusion. The model is shown here. And the take-home message from this model is simply that under these conditions, in the absence of nucleotide, it looks like FeOA doesn't make any interactions ultimately with the N-terminal domain of FeOB. Of course, this, like all models, is just a model. So Alex then used small angle X-ray scattering to characterize whether there were credence to this model. And she could show using SACs that the protein appears to be elongated as well as dynamic in solution in the absence of nucleotide. She could generate this low resolution ab initio envelope. We were very excited when we saw this because we thought, hey, that nubbin, that's got to be FeOA. We then fit our model into that. It fit pretty well. And like this cartoon here with this dancer with their leg up in the air, FeOA appears to be splayed away from the body of NFeOB in the absence of nucleotide. Now, what happens when we add nucleotide? Well, you see dramatic differences in the in-solution profile. So if we use a GTP analog that's not hydrolyzable, GMP-PNP, or GDP, we can see dramatic changes in the solution state profile of the protein, with our most compact form being in the presence of that GTP-like molecule. Now, we were fortunate that Alex is part of our chemistry biology interface training grant program. She was able to then undertake cross-training where she learned how to do hydrogen deuterium exchange mass spec. And she could use these different nucleotides and then map the different areas of the fusions to see which parts get protected or deprotected under various conditions. In the presence, in particular, of the GTP-like molecule, we see a strong and dramatic amount of protection, including that of FeOA, which is wholly consistent with our in-solution Sacs for compaction overall of that protein. So we got really excited about this. We've got modeling, we've got Sacs, we've got HDXMS. It's all showing a GTP-dependent protein-protein interaction. FeOA has to be a gap, right? Right? No. Of course it wouldn't be that simple. In fact, this was work that was done by a former postdoc in my lab, now Assistant Professor Janae Baptiste-Brown, and she used NMR to characterize the hydrolysis of GTP for these fusions. And she could show that this actually had the worst catalytic efficiency and the worst KCAT value of any of the NFeOBs that had been characterized to this point, which is completely opposite of what we were expecting. And that left us, of course, scratching our heads until we started thinking, maybe we're considering this the completely wrong way. Maybe FeOA is actually protecting the protein from getting the GTP being hydrolyzed. So Janae was easily able to answer that question. She could simply lop off the FeOA, make a brand new construct, express and purify it, then recharacterize it. She could show that it would increase the affinity for GTP, it increased the catalytic rate, and increase the catalytic efficiency when FeOA weren't present. So it appears that FeOA is actually protecting GTP from being hydrolyzed. Why? Why could that be? Well, this is a current model that we're working with here. And we think that this is because FeOB is a channel for iron 2 translocation using a monomer gated by the presence of FeOA and the presence of a triphosphate nucleotide. So under conditions in which the protein's in the apo form, there's no nucleotide bound, we believe that the transporter is off, and there's plenty of in vivo evidence that suggests that's the case as well. Then when GTP binds, that opens a channel and iron 2 can be translocated. If FeOA is present, that can stabilize the GTP-bound form of the channel, keeping the channel open. Now, that doesn't mean that the channel will never hydrolyze its nucleotide. It does so, it just does so very slowly. This is where we think FeOC could then be playing a role. For organisms that have FeOC, this could be used to help dislodge FeOA away from that of the N-terminal domain. That could increase the rate of GTP hydrolysis, and this would turn off the channel. And in fact, as far as we can tell, based on HDXMS as well as NMR, FeOA and FeOC appear to have mutually overlapping binding sites on the N-terminal domain of FeOB. But again, this is just a model. So we're working on testing this right now. Current graduate student in my lab, Candace Armstrong, in collaboration with Gabriel Maloney's lab at UT Dallas, we're currently using liposome-based assays to look at real-time transport of iron 2 then into these liposomes. We can put FeOB into these liposomes, as well as encapsulate fluorescence reporters, typically turn off fluorescence reporters, and look at how rates change based on mutations in that sulfur-containing channel, as well as how rates change based on the presence or absence of different proteins. So far, Candace has very nicely shown we can get a couple of these FeOBs very cleanly into liposomes with high efficiency. We can also encapsulate these small accessory proteins like FeOA and FeOC. And while this is very preliminary data, we are very excited that for the very first time, we've observed direct translocation of iron 2 via FeOB, and it is wholly dependent on FeOA, and it is wholly dependent on the GTP. If you have GDP there, you don't see transport. If you don't have FeOA, you see very small amounts of transport, and that turns off. But there's so much more here to do. And Candace is also part of our CDI training program. So actually next month, she's traveling to Dallas to work more with Gabriel's lab to do a whole boatload of these liposome-based transport assays to interrogate this process. Okay, so that tells you a little bit about what we imagine for the FeO system. And what we know, taking a bird's eye view, is that this is driving iron 2 uptake in many, if not most, bacteria. Unsurprisingly, it's under transcriptional regulation of iron-responsive transcriptional regulators, but this is all regulating intracellular iron homeostasis. So in the last few minutes that I have, I'm going to tell you a little bit about what we've been working on recently, thinking about how bacteria actually sense the presence of iron 2 extracellularly and respond accordingly. Now, for most bacteria to be able to sense the presence of extracellular signals and to be able to change or to signal that presence, they use two-component systems. These are composed typically of a membrane protein that has a sensorhistidine kinase sensing domain that can bind and sense varied environmental stimuli. This activates then a cytosolic domain, ultimately for ATP hydrolysis, self-phosphorylation, and that phosphate signal then gets transferred typically to a cytosolic response regulator that can affect a signal output the other end. Those signals can be wide and they can be varied, including metabolism remodeling, virulence factor production, as well as nutrient acquisition pathways. And we've gotten very interested in a specific two-component system known as BQSRS, originally identified by Diane Newman's lab in Pseudomonas aeruginosa. Now, this was based off of transcriptomics, where they noted that this two-component system is responsive to iron 2, but it's specific. It didn't respond at the transcriptomic, at the transcript level to other divalent metals or iron in the +3 oxidation state. Moreover, it's incredibly important because it affects the ability of Pseudomonas to form biofilms, and it also activates Pseudomonas against cationic stressors like cationic antimicrobials. So it's really important for its ability to become antibiotic resistant. So we started wondering, how does this system select at the mechanistic level for this cation, and whether or not it might be involved in regulation of iron homeostasis. So this project was effectively spearheaded by a single graduate student in my lab, Alexander Paredes, who's an HHMI Gilliam Fellow, who is also defending his thesis in December, if anyone is looking for a really fantastic potential postdoc. And we decided we're going to go after the whole intact system, because in the literature, these histidine kinases are typically cut up and nobody studies them intact, which is unfortunate because there's information from one side of the protein to the other side of the protein that has to be transmitted. So he first cloned, expressed, and purified the intact PA-BQSS membrane protein. He could show using analytical SEC as well as mass photometry. It appears to be a dimer in these various detergents, which is consistent with the paradigm that these function as a homodimeric unit. That has allowed us to get or to generate this AlphaFold model here on the left-hand side. And we first got interested in particular in this extracytoplasmic domain, which is speculated to be the location for iron 2 binding. So the first thing that Alexander did then when he had the intact protein is he did anoxic metal binding to try to characterize, does the protein bind to iron? And in fact, he could show using the wild type intact protein that indeed it did, with a half an iron per polypeptide, which equates to a single iron binding site per dimer of BQSS. He then was able to test whether there are certain residues that are critical for this sensing, and he could show that there's a particular glutamate, glutamate 48, that's absolutely critical for iron binding. If you get rid of that, no iron binding whatsoever. We were able to structurally characterize this using X-ray absorption spectroscopy, again in collaboration with Kelly's lab, where we could show that this binding site in the extracytoplasmic domain is a hexacoordinate binding site. We've got 4 ligands that are provided by glutamate 48, and we believe that a neutral glutamic acid is responsible for the other 2 coordination to the iron in the +2 oxidation state. And I'll return to that in just a moment. But we were really excited because, first, this is the first example that an intact histidine kinase has been structurally characterized with its cognate sensor. And second, because of the implication then for function of the histidine kinase. So if we return back to that AlphaFold model, we look then in that periplasmic domain. Here's that key glutamate 48. Here's that other key glutamate 48. If these are to come together in order to bind the iron 2, you would need a pretty significant conformational change that would have to occur in order to accommodate that. Now, that may seem like something extreme, but for the very few intact histidine kinases that have actually been structurally characterized, it appears to be critical. So for example, for NARQ that binds and senses nitrite as well as nitrate, binding of those anions causes conformational changes that then is propagated all the way across the lipid bilayer, ultimately to activating the domain within the cytosol. So then we ask the question, well, what happens when we try to look at activity of the protein in the presence of iron? And first, because we're working in a detergent micelle, we had to make sure that there weren't any contributions directly to just the cytoplasmic domain. So Alexander Clone expressed and purified the cytoplasmic domain. He showed that on its own, it's a competent ATPase, but only when you have the intact protein and only in the presence of ferrous iron and no other transition metal or no other alkaline earth metal do you see significant stimulation of the activity within the cytosol. Additionally, he was able to test those residues. What is the importance of those residues in the periplasmic domain for binding and for sensing iron? He could show that, of course, metal binding is critical. If you remove the ability of the protein to bind iron 2 in the first place, there's no activity. But geometry is also absolutely critical. You get rid of this glutamic acid, E45. It still binds iron. We've characterized it with X-ray absorption, but it has a different geometry. No stimulation. Furthermore, this asparagine 49, appear to be important for hydrogen bonding at those alpha helices at the dimer interface. You get rid of that, you also see no stimulation. So correct metal geometry, as well as the intact signal cascade, are all essential for function. Now, the purpose of this ultimately is to get a signal into the cytosol to then change the activity of the response regulator. So Alexander also characterized the response regulator, which for this system is known as BQSR. It was predicted to be a typical OMPAR-like response regulator. These are really common gram-negative response regulators. They generally have 2 domains, one that's a receiver domain that gets the phosphorylation signal, and then a DNA binding domain connected via a flexible linker. So he decided he was going to characterize that. We wanted to get structures of that protein, and he purified it beautifully. He got beautiful crystals. They diffracted beautifully. Story again, just one domain. Another example of a flexible linker in solution, you get spontaneous proteolysis. So we got the structure of the receiver domain first. As a typical beta alpha 5 receiver domain like architecture, we can identify the aspartic acid that gets phosphorylated. So we had to figure out the structure of the DNA binding domain differently in collaboration with a current postdoc in my lab, Dr. Harvinder Singh. They used NMR. They structurally characterized the DNA binding domain, typical helix turn helix motif. It looks different than FEOC, but it has a typical DNA binding domain for this type of protein. So then does it bind to DNA? Well, the paradigm here is that phosphorylation of the response regulator translates to dimerization. The dimer then goes on to form a DNA binding complex. And so Alexander prepared a variety of different species. He's got the wild type form of the protein, that remains monomeric. He's got a pseudo-activated form of it, a D51E variant, that also remains monomeric. He's got a beryllium trifluorinated activated form of the protein that will dimerize in solution. And then we've also got the C-terminal DNA binding domain. Does it bind to DNA? Well, the heavy lifting here again was done by Diane Newman's lab, where in PA14, she identified what the putative BQSR box sequence looks like. It's this tandem repeat of this TTAAG species. And she could show using a synthetic piece of DNA, indeed, you can get BQSR to bind using electrophoretic mobility shift assays. Alexander then looked into PA01, which is a more common laboratory strain of Pseudomonas. And he found that indeed this sequence appears to be upstream of a number of genes that are involved in biofilm formation, as well as our favorite iron 2 transporter, the FeO system. In fact, that system's regulation in Pseudomonas is quite complicated because not only does it have an ANR binding site, the homolog of FNR, it's of course got a Fer binding site, and it also has a BQSR box that appears to be overlapping with the Fer binding site. Will any of these bind BQSR? Well, he tested that with EMSAs. Wild type protein remains monomeric, no binding. Use that faithful dimeric activated beryllium trifluorinated species. We get a very nice complex it forms. He was also able to show that the DNA binding domain on its own will also bind. It doesn't bind as well, but it does bind and suggests that the receiver domain has some sort of autoinhibitory function because in the absence of the receiver domain, the DNA binding domain will also bind to the DNA. Now, what does that look like? Well, we have crystals of that complex. They are not great crystals yet. So for the time being, we have a model because we also have some structures of other OMPAR-like response regulators bound to DNA. They appear to make these asymmetric dimers based on AlphaFold's best guess. There's this positively charged patch ultimately that's going to drive that interaction, or we thought was going to drive that interaction with DNA. And then there was this really interesting arginine residue on this beta hairpin that appeared to be not just making interactions with the phosphate backbone, but intercalating the guanidinium into that selectivity site, into the TTAAG region by pi stacking. So Alexander generated a variety of different variants. If you get rid of that side chain and just place a methyl there, you get no binding whatsoever, despite the fact that you still have that conserved positive patch. If you then put a positive residue back in that location, you do get binding, but the binding is incredibly weak by comparison to the wild type protein, and it appears to be smeary, which might suggest some lack of specificity. So that single arginine appears to be absolutely critical for anchoring the protein into the BQSR box. Now, of course, all of this so far is all in vitro. So we wanted to get a sense of whether or not there might be function in vivo. And for that, what we did was we teamed up with Mariana Petralkin's lab at Oklahoma State University, as well as a fantastic graduate student in her lab, Kenzie Hull, who will also be defending in spring and who's also looking for a postdoc if anybody is interested. And what they showed using a deletion strain of BQSR was that indeed BQSR doesn't just affect FeO, but it affects a whole slew of different genes in Pseudomonas, but not in a way that we were initially thinking. In the deletion strain, transcripts of FeO appear to be increased rather than decreased. And we thought, that's kind of strange. This should be sensing iron 2, telling the bacterium there's iron 2 to be had, upregulate FeO, not the opposite. But it was under iron replete conditions. So Kenzie then did a series of very critical experiments where she tested iron concentration, and she also used promoter assays to test under different conditions of bacterial growth whether this changed. The regulation of BQSR is actually highly dynamic. under different conditions, FeO transcripts will be up, and under other conditions, FeO transcripts will be down, dependent not only on time and cell density, but also the concentration of iron 2 that's bioavailable. Now, how is it possible that this regulation could be so dynamic? Here's what we think as of right now. We think it's because BQSR is not a typical OMPAR-like response regulator. It seems like that ostensibly, but Alexander noted a couple of potential metal binding motifs, including a CXC motif in the DNA binding domain, as well as a histidine-rich motif, looks a lot like the calprotectin iron binding site at the interface between the receiver and the DNA binding domains. So could BQSR then bind iron 2? Well, of course, I'm leading you on to the right conclusion that indeed it does. It binds a single iron per polypeptide now, so different than BQSR that dimerizes and binds iron at that dimeric interface. We get a single iron binding site. Again, working with Kelly, we were able to characterize this using X-ray absorption spectroscopy. We could show that the CXC motif makes no difference to iron binding. In fact, it appears to be a histidine, oops, sorry about that, a histidine-rich motif at the interface between these domains, which we think is cool. And to our knowledge, it's the first example of an OMPAR-like response regulator that binds the same ion that its stimulus histidine kinase also binds. Okay, so this is a current working model we have for this system. Under low iron conditions that you would expect at the host pathogen interface where this is withheld, it's a canonical two-component system. Iron 2 binds to the S protein, signals across the lipid bilayer, phosphorylation occurs, phosphorylase, dimerization, upregulation of a variety of different systems, including FeO. Then, once iron 2 is sufficient, especially within the labile iron pool, we've got a different model here where BQSR can bind iron 2. Maybe it can also bind other metals. We're testing that. This will alter structure as well as the ability for it to interact with DNA, causing a different kind of regulation. If we zoom out, we now know, though, that these two systems, which didn't seem initially to be linked to one another, actually intimately linked in Pseudomonas iron homeostasis through their ability to sense and to transport iron 2. But we think this is going to be more globally important because there are other pathogens that have this two-component system as well, like Vibrio cholera, for example. So we're interested in also searching for this BQSRS system in other bacteria to test this mechanism. Okay, so I think I'm almost out of time here. So I've thrown a lot at you today, but just to recap those important points, what have we shown today? We've characterized and determined structures of parts of the FeO-AB system, postulated this idea that a sulfur-rich internal pathway is responsible for translocation. We're testing that right now. We've shown that FeOCs interact with FeOBs, and some of them bind iron sulfur clusters that are oxygen sensitive, but some FeOCs actually don't have that binding capability at all. We've determined FeOA regulates NTP hydrolysis by FeOB mediated through protein-protein interactions, shown this first example of iron 2 stimulation of the histidine kinase, structurally characterized the BQSR protein, and looked at this canonical as well as we think non-canonical regulation of this system in Pseudomonas. All right, so with that, I need to thank the individuals who did the work. I just get the pleasure of talking about it. This is my lab group from last spring here, as well as a fantastic group of collaborators who have helped with these projects as well as other projects. I've got to thank funding sources who support this work, as well as national resources who allow us to do especially our structural biology-based work. And I want to thank you for your kind attention, and I am happy to answer any questions that you might have.
Great talk. Questions?
Hey, Aaron, great talk. So in your FeO liposome system that you're working on with your collaborator, how do you control for the orientation of FeOB?
Yeah, so you can't effectively. So you can't control for the orientation of it, but you can control where you put the nucleotide, you can control where you put the fluorescence sensor, and you can also control where you put FeOA and FeOC. As far as we're aware, we don't know of any back transport through the misoriented transporters, but it's certainly a problem, not with just this liposome type of assay, but with many of the liposome-based assays. But we're fortunate that unlike Aporin, for example, we don't believe that we have bidirectional transport, at least of iron 2.
Right.
Seems like this should all be hooked up to ferritin or bacterial ferritin in some way. Is that something that you know anything about?
That's a good question. I do not know. Well, I should say the dirty secret is once that iron 2 gets into the cytosol, where it goes, no idea. Is there a molecule that's responsible for chaperoning it to wherever it's going next? Don't know. There are some who believe that it's just kind of flitting on and off of something like glutathione, for example, but there's evidence that suggests that that's not the case. As far as electron transport and redox as well, we know that in some systems, it is hooked up to ferric reductases. So these diheme membrane proteins that can send electrons out of the cytosol that can reduce the siderophores then down from iron 3 to iron 2 to provide iron 2 for FeOB. No, so those will get electrons from things like flavins or NADH, for example. Those electrons will go from the cytosol through the heme into the periplasm, where you've got a ferric siderophore that's solubilized. That electron will go into the ferric siderophore to reduce it down to the ferrous siderophore, that ferrous iron is a donator for FeOB back into the cytosol. But you look confused, so.
You have the system that's bringing Fe2 inside the cell.
Yes.
That's going to get oxidized as Fe2. So
No. So the electrons are not coming from iron 2. They're coming from other reducing equivalents within that of the cell. Yes. So most of these systems that we're considering, we're thinking under anaerobic metabolism type of scenario. So that it is trafficked under the iron 2 condition, under the iron 2 oxidation state. But we don't know how that trafficking occurs. And then of course, if it needs to become iron 3 in a protein, then that electron's also got to go somewhere.
But it is true what you started off with is that when the iron goes in, the idea is that if it's not used to cofactor a protein, it gets converted into ferritin. So it gets oxidized. Yep. Right. So there's a direct relationship between how much iron the cell is experiencing and how much ferritin it stores.
just because I'm confused doesn't mean
So an interesting feature, if I understand correctly, is that in this context of this discussion is that essentially because of the manner in which iron is involved in the regulation of the transcriptional regulation, it's actually then preventing the expression of the FeO system, which is then slowing the influx of the iron 2, presumably. So you have a complex mechanism for controlling how much iron 2 is in the protein, it's in the cell. Is that what I'm understanding correctly?
Yes, that's correct. And the reason why I think this is such an attractive model personally is that BQSR targets FeO and just a few other iron responsive genes within Pseudomonas, including ferric reductases. So it's a way to specifically control those systems. And then once you reach an appropriate level of iron 2, then you can have larger or more global control through FNR, ANR under anaerobic conditions, or maybe FER under aerobic conditions.
So I have a question about a second question because of just about that arginine and that really interesting apparent specificity that's being contributed by that arginine. And do you have any instinct about, this seems rather different than other UMP-R type regulators?
So is it really rather different than other UMP-R-like response regulators? They all have a beta hairpin. And on that beta hairpin, they have other residues. Some of those residues are histidines. Some of those residues are lysine. So there's a little bit of diversity in terms of what that residue is. But then some of those residues are negatively charged, presumably at physiological pH. So there's some carboxylates, but maybe the local pKa of that particular residue is tuned so it becomes actually neutral. I have no idea, but I do not know how to explain that other than if that remains a negatively charged residue, it has to be pointing away from the DNA, not into the DNA. But we were very surprised to see such a strong impact of just that one residue on binding. And we're hoping that the crystals will pan out to either say that's true or say that's complete rubbish.
Questions in the back?
Hi, that was a really interesting talk. I'm just really curious about this accessory protein that's potentially stimulating the hydrolysis. So for the systems where you don't see any FeOC, are you concerned that there might be a component that's not in the operon that might share some functional redundancy with FeOC? And do you have any thoughts on how you're going to be fishing that out?
That is a very astute point, which is there are other bacteria, and one example is Helicobacter, for which its operon does not contain an FeOC, but its genome does. And I am not a smart enough bacteriologist to figure out this question, but we work with some really fantastic bacteriologists who have been able to suss out those types of questions. Is it that there's some other FeOC-like protein that's somewhere else in the genome? That has been the case for Helicobacter, so it may be that it's the case for other bacteria. But we have another protein I didn't talk with you about that appears to be mutually exclusive with FeOC, and it shares some features with FeOC, binds a 4-iron 4-sulfur cluster, but it's a single pass transmembrane helix. And so we think that feature is more, that structural idea, or that, I shouldn't call it structural, that mechanistic idea is more conserved than we give it credit for, but in a different way by potentially using a different protein or having FeOC somewhere else within the genome.
Great. Did you want one more?
So where's the weak link that you could attack with a new antibiotic?
Yeah, absolutely. So ideally, it'd be lovely to disrupt A-B interactions. That is not an easy or non-trivial thing to do, disrupting protein-protein interactions specifically. What I think is a potential location, or a couple of potential locations, are if we go here, I think that a potential location is this periplasmic loop, which is not shown here, but you have to imagine that there is a loop from FeOB, highly negatively charged, sticking out into the periplasmic space. That is where I could imagine you could make some really interesting synthetic compounds, potentially try to get them to bind or to complex in this area, plugging up the hole, basically. The other possibility is here at the BQSRS. If you can up-stimulate this if you can upregulate the BQSS protein, we know that actually that translates to biofilm dispersal. So if you could get a molecule that binds here and upregulates that histidine kinase, biofilm dispersal is then going to make those bacteria more susceptible to antibiotics or some sort of antimicrobial. So the weak link, if you want to call it a weak link, is going to be somewhere here in the periplasmic space, I think.
So the one sort of last comment I wanted to make is I really like the BQSRS mechanism, because it's, on the one hand, iron uptake is the strategy of people, as people have investigated and think about it in bacteria, is bacteria never know when they're going to get another molecule of iron in their environment. So whatever iron is out there, they're just going to take it all up. And if they don't need it to make proteins, they store it. But this would suggest that, at least in some bacteria, there is a threshold for maybe toxicity. And I wonder what happens to these bugs if they don't make the BQRS sensor system. Do they die under high iron?
Yeah, so I think those data are shown here, where Mariana's lab shows you get, so they're not dead. I mean, this is over a pretty high concentration of iron too, right?
Mhm.
They're not dead, but you can see in the deletion strain, they have pretty impaired growth by comparison.
Mhm.
So
Yeah, so I think that makes perfect sense. And then are the nuances of VOA, VOC really helping different bacteria to have these fine tuning responses based on what their real ecosystem is, not in the lab. So I think that's really worth thinking about.
And I think generally we see FVOC more commonly with what I would argue are more virulent strains of bacteria.
Mhm. But also facultative.
Right.
Yes.
So,
Yeah.
but I hesitate, given this really important point that's been brought up, I hesitate to jump and make that link
Yeah.
necessarily, because it could be hiding
Mhm.
somewhere else in the genome.
Yeah, but the oxygen sensitivity could be linked to whether or not it's a facultative organism or not. Because then that's a whole nother problem when cells are, you're growing anaerobically, you're in an anaerobic environment 1 minute, and then you're exposed to oxygen. Maybe you need to shut down iron transport right away.
Yep.
Yeah. Yes, great. Well, great talk. Thank you very much. Thank you.
Thank you.